Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Good morning, everyone. I'm glad that Dan put me as the second speaker this morning. It's much better than when I just had a month ago. I was the second last speaker in the last session on last day of the conference. Plus, that's a Saturday. You imagine how many attendees will be there. But actually, it turned out to be quite good, and a lot of attendees stay there and listen to my presentation. Dr. Hay just mentioned about the cyber infrastructure for scientific researchers, and you probably heard a lot of talks last day about the touch upon a lot of them touch upon the biomedical applications of how to process data. Again, here I'm going to talk in the same direction, but more specifically, I'm going to give you a project from a project perspective how we're going to deal with biological data set. So my topic is the gateway to biological pathways. It's about how to put all the biological pathway data together, so make it web accessible, and also we're going to use enable this to be semantic web accessible. It's not working. Well, looks like the gadget is not working. I have to switch over. No, nothing works. Okay, I just stay on this side anyhow. Okay, here's my agenda, and I didn't steal this from Simon's slides because he had more questions than I have. Those are WH questions, but I had this in my mind before I create a slide. Those are the questions I'm going to address, why we want to do this project, what we want to do, and how we're going to do this, and what's the future direction about this project. Now, I came to several folks yesterday, and they all asked me about what I'm doing. Okay, I told them that, well, I'm working on biological pathways, so the next question is, what is biological pathways? So, in a short sentence, it is our current understanding of biological processes, okay? I'll show you how this knowledge is going to be presented. Now, think about the way that we can get the biological data, biological pathway data. First, probably a lot of them actually is curated from the scientific literature. And also, with the high-throughput lab technologies, you have microarrays, you have mass spectrometers, and you generate a lot of data. And with those set of data, you can use a computer program to infer the biological pathways. But again, those are pathways actually hypothetical. You cannot 100% trust those data, but you have to think when you use those set of data. Well, what's the use of biological pathways? Well, Dr. Kong mentioned last night. Actually, it's one of the in the NIH roadmap, and actually mentioned that biological pathways as well as the networks are very important for biomedical research. And you can use this to formulate hypotheses, and you can use this to verify lab results, and also, very importantly, you can share research outcomes with other, scientists in the field. Now, this is a biologist's view of pathways. Okay, now, if I give you how the biologist will think this pathway is, this is how it looks like. I just found this on the web and I just put on my slides. And I know it's hard to read. Probably it's better than physicians' prescriptions, right? And those signatures are very hard to recognize. But here you can still see the the lines, edges, and different notations, but the different components that involve in the biological pathways. Well, thanks to the technology now, you can draw the pathways this way. So this is, I believe, it's from CAC. Database, and you'll see that it's quite overwhelming because you have so many items that involve on this diagram, and you'll figure out the different kind of relationships. Now, this is a different way of representing the pathways. It's from BioCyc, the website hosted by the Stanford Research Institute. You'll see that even though we are talking about biological pathways, but when you talk to different people have different way of representing the pathway. As one of our fellow folks from Indiana, Memo, he mentioned that yesterday about the different ways of representing this pathway data. So it's kind of interesting field in the bioinformatics field. Now, well, how many databases are there? Well, there's a list called a biological pathway resource list, and it's over, right now, there are over 180 pathway databases. Well, compared to a lot of other databases, still very small fraction of the entire biology databases. Now, well, those databases all start with different interests, different focus. Different labs may have different interests on specific projects, and they all have different representations, and they all use different storage formats, and also they all use different access methods, and well, coverage will be different as well. Now, for biologists to address any intriguing questions, a lot of time they have to query multiple databases and in order to get some answers to their questions. Well, well, that's a very common practice for them. Now, the challenge is they don't have any unified interfaces for querying the databases, and they have to go through different databases and use different ways to query the databases. And plus, most important thing is that right now, everybody has web presence. Well, that's good. From older days, you all have those text-based, database you do query. It's kind of very challenging for biologists. But now everybody has web presence. Oh, I think that's a good way for biologists. It's right. It's very easy for human eyes to use the web. But in terms of let the computers to process this, everything embedded in the HTML documents are going to be very difficult to process, because HTML pretty much is the standard for it's a protocol for formatting the content, not really for composing or storing the content. So it's not machine processable. Now, I'll give you a scenario here about how to use different data sets in biomedical research. For example, if a biologist is interested in some enzymes in a metabolic pathway, he may find that those enzymes actually are translated from some genes. Okay, now. And then later on, he may find that, oh, those genes actually are regulated by some transduction factors. Well, further search may indicate that those transduction factors may be triggered by some cell surface receptors. Now, look at it here. I have 3 sets of pathway data, and they are related. This is a challenge that a lot of biologists will have, and how to go through the search process to find the right information they want. And one of the speakers mentioned yesterday that for scientists, they experience a problem of information explosion. They have so much information to process, and they have too much data to digest, to absorb. So what I'm going to do about this project is I'm not going to integrate all those data sets together. It's going to be very challenging for us to do this. Now, well, a lot of data is out there. Okay, a lot of data sets out there, but the challenge is no unified interface to access data. Well, second is this relates to biological database integrations. It's everything we have had gene bank or other databases, and the database integration has been very challenging research area for a lot of folks out there, and they try so many different ways to achieve this. I think we're still not there yet. Well, one way to do integration is to develop some common schemas. I know it's very challenging for different research group have different interests, and also different domain have different way of representing knowledge. So it's very difficult to do this. And our fellow folks at the IIU memo mentioned yesterday that when you do integration, you should not do this on by syntax. Okay, you have to do this at semantic level. You have to do this by meanings. Okay, otherwise you're going to end up with something that's not necessarily useful for biologists, for biomedical researchers. Now, so this project is about creating a portal for biological pathway data sets. Okay, now what we're going to do is we're going to support the semantic web. There are a lot of talks about the semantic web in life sciences. W3C actually had a workshop last year about semantic web in life sciences. A lot of them actually, that researchers study, nobody really had some actual data you can access online. Because I mean, when you talk about semantic web, you talk about the web of data. I mean, when we talk about web, we talk about World Wide Web, right? So I mean, if everything is stored on your local hard disk, well, this probably will defeat the purpose of semantic web anyhow. Now, we're going to use BioPAX format. I'll get into BioPAX a little later. BioPAX actually is a standard for common pathway format. Okay, I guess I'm lucky to find some common common ground that everybody can agree upon with. And I want this to be accessible from through the HTTP GET. If you are familiar with semantic web, you probably noticed that every thing that described in the semantic web actually will be web resources. So how you can access the data? So I'm going to the best way to do this is use the HTTP protocol. And with semantic linkages, the data we present here probably will be able to link to different external databases. Now, and I also want to support the XML server-based web service. I know I should not put the XML server, but when you talk web services in the bioinformatics landscape community, and you'll notice that not everybody will have the same definition of web services. Okay, when we say the web services, it can mean different things. So I did this on purpose. I said it's XML server-based web services. And once we provide the data, and then bioinformaticians from all the labs can just create their own rendering, and you'll see there's so many different ways to present the pathway data, and they can do whatever they want to do. And another important thing is we try to let different platform implementations to consume the data as well, because a lot of labs out there in the bioinformatics field are using Apple, using Linux, Unix, but so they'll still want to access the data. And also important for us is to provide a unified interface for querying pathway data sets. And this project is not going to take care of the data cleaning issues, because this had a lot to do with verifying all the data from the experiments, and also we're going to generate any new data in this project as well. Now, so what is BioPAX? Okay, BioPAX is the is a standard that developed by a group of researchers in at a university, as well as the pharmaceutical industries. Okay, those folks actually are the experts in the field of biological pathways. They created a consortium called BioPathway Consortiums. They defined this project. It's been several years, I think about 2 or 3 years, ongoing process. They have very good discipline in keeping up with monthly meetings, and then they publish minutes online, and you can go there and take a look at what they do. And and they define about BioPAX ontology. Okay, it pretty much just define the entities in the biological pathways as well as their relationships. They had a vision about semantic web, and so they created this using OWL. It's a Web Ontology Language. That's it's also the 3WC recommendations out there. You can take a look out there. And well, another interesting thing about this ontology is that it's in XML format. Okay? Unlike a lot of ontologies, it's just this texture descriptive document about the ontology, but this one is XML document, so you can pretty much use it to process that. You can use it as a schema to validate a lot of documents. And this is the levels of biological pathways. They cannot accomplish everything at one time, so they have to do this by levels. First level is metabolic pathways. It's officially released last year, and I think last year or early this year, and it's out there. You can use it. They have a website out there. You can just point to the URL and look at the ontology. Also, you can download it on your machine in case you want to do some local processing. And the second level actually is still in candidate release 2, and it's already on their web, and you can take a look at it. It's about molecular interaction networks as well. Level 3 is about signaling pathways. Also, they have 2 more levels on their roadmap, and you can take a look at their website. Now, this is how the BioPAX ontology will look like. Well, you probably were familiar with object-oriented program. This looks like the URML for the BioPAX ontology. And on top, you have entities, and this is they call root class, and actually everybody inherits from the entity, and you have different kind of interactions. Also, you may have a pathway, and you may have some physical entities. Can be a complex, can be RNA or small molecules, the proteins, and among other things. Also, here you probably noticed that they don't just define all the physical entities, they also define those abstract entities as well, like reaction. It's a process, it's not necessarily the physical entity you can touch, you can feel. And I didn't show you the other part. Another part actually is called utility class. Actually, is another set of classes that relate to the biological pathways. Here is an example of what you actually see in pathway class. So you have those, they call it slots, or sometimes they call it properties, but in object-oriented program, you probably call it attributes. Those are the attributes. Those are the common attributes inherited from entities. Those are the specific ones that for the pathways. So those are kind of things they already defined in the ontology, and you can use this to validate your the data. So here's what we propose to do. We have a system that will process the data, that will store the data internally in BioPAX format, because we don't want to do conversion or transformation one more time. So, and we also have the query engine that will retrieve the data from our internal storage. And well, there are several ways to get data. You can get data from those Haskell database hosts, and you can download it from there and just process. We have data source components. They actually process different set of data. And also we thought about using the web services. CAG provides web service to access their pathway data, but not in the BioPAX format. And we use BioPAX ontology to transform the data from the sources and then storing our internal persistent storage. This is where we store the BioPAX format data. They are in XML format. And we also provide have a query engine that will query the storage, the data stored in the persistent storage. And also we have web services that everybody can query. Our HTTP access method actually using the web services to accomplish this. And then we can create all kinds of client applications with our group can create, or with other interested bioinformaticians want to create it. They can also access our web services to retrieve data and create their own application to run their own. Oops, I skipped too fast. Sorry, I have a few more slides. Yeah. Sorry. Now. This is what we do with our internal data store. Pretty much everything is in XML format, and well, larger data set compared to the others, of course, it's relatively small. But when we talk about larger data sets, for example, if you look at a data set from BioCyc for each organism, and you have a very large XML document, and we try to process that file, and with different kind of technologies, and none of them really work quite well because. The issue is about the memory, because a lot of XML parsers cannot hold that much data into the memory. And we thought about different strategies to solve the problem, and one thing came out was from my experience with a technology Microsoft developed called a web storage system. in the XQuery Server 2000. I don't know how many of you are aware of this technology, but actually the technology was state of art, actually was ahead of time, at time that everything you could do with web storage system using XML, even though it has the ADO components out there and you can access, but what's behind it is in XML format. You can create a schema, you can create the file structure, you can do everything using XML. So I had this idea, and then went to the tech ed and met Michael Reese. I don't know whether he's here today. I sent him an email yesterday, and I hope. Oh, he's in the back. Yeah, good. We had a talk at the conference, and I just want to know about the XML support in XML in the SQL Server 2005, and we had a chat, and he invited me to his presentation the last day of the attack. Actually, I learned a lot from him, and we had some email communication going on about how to solve our problem. But anyhow, we look into the XML data type in the SQL Server 2005. It looks like it's a very promising technology for us, and our job is how to put data in there. And with the XML data type, what you could do is you can put the different kinds of XML document. You don't have to have the document with the same schema. Okay, I'm talking about in the same same column. So that's the beauty of doing that, because we can simplify our query queries. So, and we just decided to put those small entities in the XML data type column. We have paths, we have small molecules, we have RNA, then we have proteins. We can put all this in the in the XML data type column, and also the SQL Server 2005 support indexing on the XML data type, even though it's blob data type, it still support the index. And another beauty by using SQL Server 2005 is it supports the XML, the XQuery. Michael is the expert on the XQuery, and he wrote up a lot of articles and books on this XQuery. So we're using the XQuery technology for our application as well. And the in order to get different entities, you can just call the you can just perform multiple queries to get those entities. And actually, this really helped us to eliminate the complexity of design very sophisticated database scheme. Otherwise, you have to use different technology to achieve this. Now, it's working now. This is how the one of the pathway entity will look like in the database. It's pretty much like this, except that we have namespaces for all this, because we install the data, XML data in the XML data type column, you have to have namespace there. So this is what you look like, even though you don't see the all the low level details out there, because like for example, pathway components actually is made up of interaction steps, and then you can look at different the participants in the interaction steps to find what kind of what kind of biological elements components are involved in the pathways. Now, this is our website out there. I wish I could do some demo, and after I talked to Dan, and I decided not to do. The challenge is that I have to use non-Microsoft technology. It's not just something that we did with Microsoft. It's that little glitch in the Microsoft technology that we have to use the Firefox. I don't think this laptop has a Firefox to do this. But this is our website. You can go there and take a look, and we have quite comprehensive documents and about how to access our data sets there. And you can use web services. You can use HTTP GET POST methods to access our data. And again, like any other Microsoft web services, you have a visual file. You can just consume the way you want. And for those who are not familiar with web services, we provide documentation on how to use this with Java, with C#. A couple of my students actually did an excellent job on this. They spent a lot of time writing the documentation. And now, what about future? Okay, well, we are providing data now, but we don't have any way to consumer data to visualize data. So our next step will be writing a client application to consume the BioPAX format data. Our initial goal was to create something in C#. Well, the reason for this is that we are thinking about the large bioinformatics community. A lot of them using Mac, using using Linux, Unix. You all know that C# runs on those boxes too? No clue?
Yeah.
Oh, yeah, Mono project. Actually, yeah, Dr. Ray, thank you. Yeah, he's the only one who knows the answer. That's why I was thinking using the, unfortunately, Mono project actually has not been able to provide support for WinForm class. So we'll not be able to do this at this moment. I think I will probably just, go away with our initial goal. So we'll pursue a different approach to do that. The linkages to external data, it's already there. All you have to do just will find how to access different data sets out there. For example, GenBank, actually Entree has a e-utility. It's in web they provide web services. You can access their data sets. So any literature, any reference in the pathways actually can be accessed from the Entree e-utility web services. And also, we're thinking about how to update the data sets from our external data sources. And last two probably will help us to do semantic data mining as well as machine reasoning, because everything is in the machine processable format. All you have to do is just write a code to process the data and write a code to process the data. I'd like to thank Microsoft Research for support of this project, and also I'd like to thank Dan for all his work to provide all kinds of support that we need for this project. Our collaborator, Dr. Shun from biology department of our campus, and actually he's working on radio frequency effect on human cells. Actually, it's a very interesting project. It's sponsored by Air Force. What they are doing is they are conducting research on how the radio frequency can affect our human. And what they use, the radio frequencies they use actually are the ones that the cell phone industry uses. So you'll think that they'll have potential impact on how cell phone will affect our health. And one important thing they are looking at is they're looking at different pathways. So as one of the pathways is apoptosis, and they know that they try to find out whether any radio frequency will affect cell death. And also they have so much data from their microarray experiment. They really want to look at different pathways, how those cells, those genes, different biological components play roles in the to the external disturbance from the radio frequencies. And also I'd like to thank my students who worked real hard on this project. And I'd like to entertain some questions. If you're shy to answer questions, you can send a question to me through this email. And again, this is our website, and I really encourage everybody to go there and take a look and consume our data and give our comments back. And we'd like to see more use of this web portal.
Thank you.
Just set up all your. No question? Any questions?
I have a geeky question, which is how did the XML stuff work out? You're kind of vague about it. You said that it was experimented with it, but you decided that it worked.
Well, actually, we tried Jenna first, okay? Jenna is the one that we tried, and I mentioned that the XML parser issues because we cannot handle the large amount of data because the way that initially we look at the BioPAX, BioCyc data, and it's in large XML document, and there's no way you can load into the XML into the memory. Yeah, and also we look at the Symphony, and one of my students.
Did the SQL stuff work?
Oh yeah, SQL stuff. Yeah, the SQL stuff works quite well. It's just beautifully, and we tried the system as well. Yeah. Okay, sorry. Yeah.
would you like to comment on the usefulness and applicability on the, for example, the signal transduction knowledge environment that's produced by Science Magazine? Because they put forward a set of tools.
Yeah, actually, this is a level 2. One of the group actually already working on the level 2 data, so that hopefully before long we're going to import this into our system, so it will be provided to the bioinformatics community.
Right. So you're just surveying all of the various different databases out there and bringing them all in no matter what. They have one of the nice things about their design is that they have a way of diagramming building signal transduction hub.
Right. Yeah. Yeah, we can do that with our client. It depends because as Memo mentioned, that there are so many different ways to run the graphic representation of the pathways. It depends on your research focus. It depends on how you're going to solve your problem.
Yeah, but the point is that if you're interested in standardizing that, then a visual nomenclature that is actually being used by the community could be useful for you.
Right, I agree with you. You have a very good point. It's another community effort to standardize this. I think it's not there yet. Okay. Oops.